So in laboratory settings, it's a very process. So what which is fine. I guess the price. The difference. Like you said. Yeah, it's all like
Hello, everybody. We're going to go ahead and get started with the Biotech pitch contest. I'm Jennifer Beach. I'm Associate Director of the Blavatnik Fund. The 7 pitches you'll be seeing today are competing for a $30,000 Blavatnik Accelerator Award prize. They were chosen from the 13 e-posters that you guys had seen outside in the courtyard. If you guys are interested in these or learning more about the Blavatnik Fund, please come see myself and Bill Wiesler after this. So I'll hand it over to Rob Bettigole, who'll be moderating the session.
Hello, everyone. I'm Rob Bettigole from Elm Street Ventures, venture capital fund, right down Whitney Avenue from here. There's going to be very little between sessions. We're just going to have one question per speaker, and I'd like to invite Julie to come up and give the first presentation.
Hello? You guys hear? Yes, okay. All right, good afternoon, everyone. Thanks so much for the opportunity to share this project that I've been working on. So this is me. I'm actually a pediatrician and a nephrologist. I have an active clinical practice, but I'm an investigator in the vascular biology and therapeutics program at Yale. My area of focus is vascular inflammation, steroid microenvironments, and endothelial cells, and more recently, Wnt signaling. The problem that I'm trying to tackle is that of cardiovascular disease, which remains the #1 cause of worldwide mortality. Despite many FDA-approved drugs since 2010, mortality continues to increase, and healthcare costs are also increasing in the range of hundreds of billions of dollars. So clearly, the approaches that we have are not sufficient. Patients with cardiovascular disease have a lot of homework, a lot of modifiable lifestyle factors that they're supposed to address. And besides that, the pharmacotherapy is mainly centered around lipid-lowering agents. Our lab has identified the Wnt signaling pathway as a key mechanistic pathway in the development of cardiovascular disease through an unbiased genomic screen. However, there are no current therapies for cardiovascular disease which address this pathway. So here I want to share some data that we've generated in my laboratory. So I'm showing here isolated mouse aortas stained with oil red O, which turns red wherever there is lipid. And these mice were fed for 12 weeks with a high-fat diet, and after 4 weeks of diet, they were given a Wnt inhibitor for 8 weeks. I have two models here. One is the ApoE knockout model on the left. More. Oh, hello? Oh, much better. Okay. Hopefully you heard some of what I said. So anyway, yes, these are isolated mouse aortas that were dissected out from animals fed a high-fat diet for 12 weeks. After the first 4 weeks, they received a Wnt inhibitor, which is termed here LGK974. And there are two models here. The first is the ApoE knockout model, which is a standard model of mouse atherosclerosis. The second model is a model I generated, which is called the DKO model, which is more severe, obviously, you can see. And it's a knockout of the endothelial glucocorticoid receptor on the background of the ApoE knockout animal. And you may be wondering, why did we even attempt to use Wnt inhibitors in this model? And that was because when we were trying to understand the severity of the phenotype, we did ChIP-seq for GR and endothelial cells and demonstrated that in the absence of endothelial GR, Wnt signaling was highly upregulated. So basically, you can see with the addition of the Wnt inhibitor, the lipid deposition is decreased by about 50%. We can also demonstrate a significant lipid-lowering effect of about 50%, which is similar to standard of care and perhaps better. However, we can also demonstrate two additional mechanistic insights, which are not addressed in current therapies. The first is mitigation of inflammatory cytokines. Excuse me, my voice is very bad after talking during lunch. The first is mitigation of inflammatory cytokines, and you can see the addition of LGK974 suppresses inflammation by about 50% in the blood. And secondly, we can show in isolated endothelial cells that there's restoration of fatty acid oxidation, which is very important because defects in fuel preference have been shown to contribute to fibrosis and mortality. So currently, I've been awarded about $3 million in NIH funding to study this process. Currently, I have a patent that was issued last June. The claim set allows for the use of Wnt inhibitors as therapy for cardiovascular disease, and it allows for new compositions of matter. So this would represent a first-in-class therapeutic opportunity. And basically what I would like to do is develop an endothelial-specific Wnt inhibitor by doing a high-throughput microscopic screen in endothelial cells using several curated small molecule libraries which have been selected based on the mechanism of action of the current inhibitor. And then eventually take those leads into our validated mouse models and do comparative efficacy studies based on current standards of care. Thank you.
Do you have questions from the audience?
So for the phase 1 use, what is it about the endothelial cell types that would lend itself to some specificity for these libraries of molecules you'll be screening, Wnt in endothelial versus other cell types?
Well, I think it's not that the cell type would necessarily lend itself very well, but the fact that endothelial dysfunction is generally recognized as the first step in the development of atherosclerosis, so it would be the very earliest intervention phase, and also the fact that many of the beneficial effects we've seen with the inhibitors can be mediated solely through endothelial cells.
But these would be administered systemically, hit all the cells.
Potentially, I think one part of the development could be demonstrating the specificity to endothelial cells, and if that was not the case, there are endothelial-specific drug delivery platforms, which I think could be employed.
Great, thank you very much.
Thank you.
Peggy's already here. Microphone.
Do I just advance?
Good afternoon, everyone. My name is Peggy Myung. I'm a physician scientist in dermatology. Before I begin, I just want to make sure that we're all on the same page because it's fundamental to this technology. The same structure that gives us our hair and our teeth is the same structure that gives birds their feathers and reptiles their scales. That structure lies just beneath the skin surface, and in my lab, we're trying to recreate that structure to regrow hair. That structure is called the dermal condensate, or DC. DCs are composed of specialized dermal cells that initiate hair follicle formation during embryonic development, and in the adult, they are responsible for hair regrowth. However, the hair follicles you have are the ones you were born with, and if lost or destroyed, they cannot grow back on their own. Currently, there are no effective treatments that induce the formation of new hair follicles. The most common form of baldness is male pattern baldness, and as many of you know, many of the first-line treatments, including minoxidil, only slow down or delay the progressive loss of hair over time. Other more durable, noticeable hair treatments entail taking hair, pre-existing hair from uninvolved areas of scalp and then transplanting them onto balding areas. However, this procedure is costly, invasive, and frequently can't recreate the natural texture and density of hair, especially over the hairlines. Given that hair transplants cost over $10,000 per patient, we estimate the market value for effective novel treatments for alopecia or baldness is over $5 billion. Our approach is drastically different. What we want to do is we want to make brand new hair follicles in area of hair loss. By resurrecting embryonic programs of hair follicle development or formation. And as I mentioned to you before, the key to achieving this goal is to make DCs, hair-inducing DCs in skin. This would achieve two things. First, DC cells can initiate new hair follicle growth. And second, we can control the size of the hair follicle because we know that the number of dermal cells regulates hair shaft size. So what this means is that we can restore the youthful thickness of hair for both men and women while also recreating the natural gradient hairline so it doesn't appear artificial as what you typically see after hair transplants. However, the major barrier to using DCs to make new hair is the fact that the molecular signals required to generate DC cells in the first place have been unknown for decades. Well, that changed recently. Our lab was the first lab to identify the key signals required to generate DC cells. we call these signals signal X and signal Y. We showed that these two signals together are necessary and sufficient to form DCs in a mouse model. Further, we showed that the level and duration of these DCs, of these signals can modulate DC size and therefore hair thickness. Currently, we are at the next step of wanting to determine the dose and duration of signal X and Y that can modulate the size and formation of hair follicles or of DCs. For this, we generated a more efficient model to test this by developing the first in vitro dermal in vitro culture model that can generate DC cells using chemical agonists of signal X and signal Y. So generally what we're doing is we're taking cells from a non-DC state and then programming and propagating them into a DC state, which cannot be done with other standard state-of-the-art in vitro models. So this accelerator award would be critical for us at this dose and duration stage. Specifically, we want to use this in vitro model to test specifically the different doses and duration of signal X and Y that is necessary for optimal DC gene expression and DC expansion. And second, we want to verify and measure the ability of these cultured DC cells to make new hair follicles using a standard live animal hair growth model. We estimate the cost of these experiments will be about $28,000. These groundwork experiments will set the stage for being able to then transition into developing small molecules for human drug delivery. This could be either topically as an ointment or cream that can be applied on top of the skin, or injected intradermally, somewhat akin to Botox injections. Finally, ultimately, we expect that our first patient test population will include patients with male pattern baldness. And finally, I'd like to thank you for all of your attention and for giving us this opportunity to present our research.
I felt like I was the model standing up there. However, questions from our judges would be welcome. Not signal. Yeah, okay.
Just in terms of potential tumorigenicity, to what extent has that been explored, or just safety in general with the X and Y?
That's a great question. So I think the true beauty of this model is that once it's given, it's done. Meaning, once you make the DCs and they can initiate hair follicle formation, you no longer need continued chronic, exposure to these chemicals. The hair kind of is a self-perpetuating factory at that point. So really, you just need to make the DCs, and then they're able to induce the hair follicle, and then it's done, and you have a permanent hair follicle.
Thank you very much. Our next speaker is Prashanth.
Thank you. I see you conveniently voted my last name. Very smart.
I was practicing today.
I'm one of the faculty here in the Department of Surgery. I serve as the director of the Aortic Institute here at Yale. My topic today is the development of a novel biomarker platform for this condition in aortic aneurysm disease. Up to 5 to 6 million Americans have aortic aneurysm disease. It's mostly asymptomatic. This makes the diagnosis of this disease and condition very difficult. Aneurysms complications are quite catastrophic. They often present without any warnings suddenly, and they entail aortic rupture or dissection, which is nearly catastrophic, resulting in sudden death in over 80% of the patients who have this complication. It's often referred to as a silent killer. Once detected, which is typically incidentally done, lifelong surveillance is recommended for this condition, and surgical intervention remains the only treatment option for these patients. Current guidelines for aneurysm surveillance and decisions about ongoing surveillance versus surgical intervention are mostly based on imaging modalities, which only provide macroscopic information. To this day, we have no biomarker or blood test to help guide the management of this condition.
See the blood moving there?
So, aortic aneurysm disease, I'm sorry, I wish I could mute this.
Yeah.
My goodness gracious.
Sorry, it's from the operating room. I apologize. So aortic aneurysm disease. I'll skip to the next step. So what we see with aortic aneurysm disease is that there's huge clinical gaps with this condition because current guidelines that are based on maybe the source volume. Sorry about that. Thank you so much. I apologize. So there's a huge mismatch in what the guidelines recommend and what we see in the clinical practice. One of the major problems with this condition is even though guidelines recommend intervention for aneurysms at 5.5 centimeters in diameter, where the normal aorta is about 2.5 centimeters, most patients who present with aneurysm complications have aneurysms less than 5 centimeters, which leaves a huge clinical gap to fill. There's also no distinction for guidelines regarding the location of the aneurysm. And the biggest problem is imaging modalities are often lag behind compared to what we see with regards to progression of the disease. So what we find is that whereas imaging guidelines give us macroscopic information, we have no molecular window into the disease progression process at all. This leads to the critical need for why we need some kind of a blood test or biomarker to help monitor for these patients. Most biomarker platforms so far to treat this condition have been blood-based, of course, and they've all looked at many microparticles such as exosomes, free plasma proteins, and nucleic acids in the blood as biomarkers for aneurysm detection and surveillance. But almost all these tests have not achieved any clinical penetrance because if you take a blood sample, it represents exosomes, nanoparticles, and proteins that are released by all cell types into the bloodstream. So this just leaves a huge dilution factor that we have to account for. So what we said in our lab, and what's unique about our lab, is that we take the nanoparticles from a plasma sample and we added an extra step where we enrich for the exosomes that are released by the aortic endothelial cells. And that enables us to enrich the signal and represents the signal that's coming from the aortic endothelium over 100-fold compared to a whole blood analysis. So if you look at the aortic aneurysm wall, or aortic wall, there's two main cell types that make up the aortic wall. That is endothelial cells that line the inside, and then the vascular smooth muscle cells that coat around the endothelial lumen. We've now developed methodologies and validated this in mouse models of aneurysm development, where we've shown that when we profile these endothelial specific exosomes in the blood, we're able to detect aortic aneurysm disease and track it. We've now extrapolated that to the clinical setting, so I'll share some of that data. So we've now been successfully enriching endothelial cells, endothelial exosomes from aneurysm patients and control patients, and we completed a cross-sectional study of 40 patients with aortic aneurysm disease and matched them to 40 age-matched control patients who don't have aneurysm disease but have other cardiovascular conditions. When we looked at markers, we found significant differences. VE cadherin is an endothelial-specific marker. When we looked at VE cadherin expression in the aortic endothelial exosomes of aneurysm subjects compared to their matched controls, we found that at the protein and mRNA level, VE cadherin as a cargo of the endothelial exosomes was significantly decreased in the aneurysm patients. Another markers that we tested were what are called matrix metalloproteinases. These are a class of proteins that are very important in maintaining the integrity of the blood vessel wall, the aortic wall. And what we found with these was that these are upregulated in aneurysm conditions. I apologize. These are upregulated in aneurysm conditions. So if you look at aortic aneurysm tissue, MMPs are upregulated and they break down the aortic wall, leading to aneurysm development. Based on this, we actually looked at MMP expression in the aortic endothelial exosomes, and we found a similar pattern. We noted that in these aneurysm patients, MMPs are upregulated in their exosomal output as well. So taken together, we think that we have a new molecular window into this condition that can help guide management of these patients. The Blavatnik Accelerator Award, the project we wish to complete is a larger patient cohort. We wish to complete the cross-sectional analysis based on these markers we've already identified. Our labs has now performed next-generation sequencing and identified new microRNA biomarkers that are significantly altered in aneurysm subjects. We wish to complete that. And we're now also starting to look at patients who come in with aortic emergencies to look at these marker differences. Thank you.
Thank you. Judges. Okay, let's move on to the next speaker. Thank you very much. Susan, Baserga.
Thank you. Hello? You can hear me, yes? Where's the clicker? There it is. Alrighty. So I'm here today to tell you about how we target RNA polymerase I in the cell nucleolus. Those pink things are nucleoli, as a new approach to target triple-negative breast cancer. There's a huge unmet need for triple-negative breast cancer. It's one of the deadliest of all breast cancers. If you're diagnosed with metastatic disease, you have a 12% 5-year survival. It tends to occur in younger women. It occurs in women who are African American or who are Latina. There are no personalized therapies. So we've decided to take a new approach to target the cell nucleolus to discover new drugs that can target triple-negative breast cancer, or TNBC. This is me. I'm Susan Baserga. I'm a professor of biochemistry here at Yale. I've been studying the nucleolus for the last 30 years. I'm good at what I do. We've decided to take a new approach using our basic science expertise and turning it now to cancer therapy. This is not a new idea. We've known since the 19th century that the nucleolus drives cancer. This was the work of Giuseppe Pianese, who first observed that cancer cells had larger and more numerous nucleoli. And that brings us around to the idea that perhaps we could find small molecules that would then inhibit the most important enzymatic molecule in the production of ribosomes in the nucleolus, and that is RNA polymerase I, as you can see on the slide behind me. And in fact, we already target the nucleolus with some of the cancer drugs that are already in existence. They just weren't made to do that on purpose. And because they weren't made to do that on purpose, they have a lot of toxicities. So we think that we'll be able to use our new screening platform to find drugs specifically targeted for the nucleolus that would have less toxicity. Oops. So we've exploited a screening platform that my laboratory developed that is very simple. It just counts the number of nucleoli in a cell. Most cells have 2 to 3 nucleoli. If you disrupt ribosome biogenesis, it goes from 2 to 3 to 1, or 2 to 3 to 5 or more. We've used this screen in a number of ways. We screened genome-wide for siRNAs, also for microRNAs and long non-coding RNAs, and we've published many of these results, as you can see here on the slide. We've decided then to use this for small molecule discovery. And we screened this Life Chemicals FSP3 library, which is about 25,000 compounds. We have excellent screening statistics. We have about 200 active compounds, which is about a 0.8% hit rate. 137 of those were reproducible, and they can be classified into 11 different chemical structure clusters based on the Data Warrior software. We tested them to try to find a target, and we used an assay that was developed in my laboratory by 2 of my graduate students, Carson Bryan and Mason McCool. And it was an assay that assesses the activity against RNA polymerase I, this very important molecule in the cell nucleolus. As you can see on the slide, almost all of the compounds that we tested have activity against RNA polymerase I, shown here benchmarked against BMH21, which is a known inhibitor of RNA polymerase I out of Johns Hopkins. So we have a series of molecules that we now know the target for that we're then next going to test for their efficacy in killing triple-negative breast cancer cells in vitro. And that's what we've done here. And this is our most exciting data. This shows that the screen actives preferentially kill a triple-negative breast cancer cell line in vitro. We tested representative compounds from each of the 11 different chemical structure clusters. 10 of the 11 gave us differential killing. These are the best 3. So we just call them 1, 2, and 3. And as you can see, we compare the TNBC cell line, MDA MB468, against the near normal breast epithelial cell line, MCF10A. In 2 cases, we have differences that are as much as 2 logs, and in one case, for number 1, we have nanomolar efficiency. So we're very excited now to move these into mice to test to see if we can actually cure tumors. We have some competition. We have a group in Australia that's competing with us, and they're tumor agnostic, not dedicated to TNBC like we are. And they also target RNA polymerase I, as does BMH21 out of Johns Hopkins. But in both cases, these molecules were discovered by in vitro assays, not for the cell-based assay that we used. And we instead have a different approach, where we use a cell-based assay to discover molecules that are focused on TNBC. So I'm pitching for funds for proof of concept first in vivo tumor efficacy. All the work I've shown you so far has been done with funds from the Breast Cancer Alliance. The next step for these 3 screen actives is to test them at the Yale Center for Precision Cancer Medicine for the maximum tolerated dose in mice, and then to see if they can cure this TNBC tumor in vivo.
Well, that was perfect.
Maybe just based on the mechanism, what
Yeah.
do you think the potential toxicities would be?
So the toxicity you worry about is that it would affect normal cells just as much as cancer cells, but our in vitro data show, already that it's affecting cancer cells more. But I agree with you that we have to test it in vivo to be able to know for certain. Thank you.
Up next is Thuy Tran. There you are.
Thank you, everyone, for this opportunity. So what I'm pitching is for development of a bispecific antibody against macrophage migration inhibitory factor, or MIF, as well as D-dopochrome T-tautomerase, or DDT, also known as MIF2. So this is our team. I'm a melanoma medical oncologist by trade. I do translational research, and I work closely with Dr. Richard Bucala, who is the preeminent international scholar in MIF biology. So he was also involved in the initial early phase development of imalulimab, which is our first generation oxidized MIF antibody. So why do we care? So the prior attempts at clinical targeting of this pathway have really focused only on half of this pathway, basically targeting anti-MIF or anti-CD74 and ignoring completely anti-DDT potential therapeutics, which have not been targeted to date. So CD74 may seem like an ideal target just because MIF and DDT both signal through it, but in fact, CD74 is found on a lot of immune subsets, particularly T memory subsets, which are critical for tumor responses in memory. CD74 is also correlated with improved outcomes in oncology as well. So theoretically, maybe not the best target. Why we're focused on targeting MIF and DDT is that we know that there is synergism and additive benefits in targeting both to inhibit this pathway. Why a bispecific? It's because we can subvert some of the fallbacks with imalulimab development, meaning we're trying to minimize protein aggregation, and that we're also trying to make this a more streamlined, efficient approach to bring through early phase clinical trials. Additionally, bispecific will allow for more efficient targeting of these cytokines and neutralization within the tumor microenvironment. So MIF in oncology is bad. We know that in terms of autocrine tumor signaling, it increases pathways associated with proliferation, increases tumor-associated angiogenesis, and within the immune cells in the tumor microenvironment, it increases myeloid-derived suppressor cells, tumor-associated macrophages, as well as it increases PD-L1 expression. So in the TCGA data set, we found that high MIF levels are associated with worse outcomes to standard immunotherapies, anti-CTLA-4, as well as anti-PD-1 therapies. What we have shown is that within tumors, ovarian patients as well as melanoma patients, the protein and the mRNA levels of MIF and DDT are highly correlated. Furthermore, in our immune checkpoint treated patients, where we look at CD74, high expression and low MIF expression, so the ratio of these, so low MIF and low DDT relative to CD74 is associated with improved survival in terms of progression-free and overall survival in our patient population. In vivo data using a murine syngeneic tumor model has shown that really anti-PD-1, which is a standard of care for melanoma, only has marginal benefits. However, when we target specific subsets in this MIF-DDT CD74 pathway, anti-CD74 alone in the solid line as well as in combination with PD-1 only improve survival by a fraction. Anti-MIF alone and with PD-1, also same results. Anti-DDT as well as with the combination of PD-1, also again marginal benefit. Interestingly, it's really the combination of anti-MIF and anti-DDT which led to robust overall responses in our animals and tumor shrinkage as well as prolonged tumor memory. This is also synergistic and additive with existing immune checkpoints, suggesting that anti-MIF and anti-DDT targeting is a novel therapeutic checkpoint pathway that can be utilized and is synergistic with existing immune checkpoints. This also represents a very tumor-agnostic approach because many tumor subtypes have high expression of both MIF and DDT. Just to reiterate, we have not made significant progress in this field beyond ipilimumab and nivolumab since 2015. So a lot of the current therapeutics that are being approved are Me-Too monoclonal antibodies targeting the same proteins in this checkpoint cascade. So MIF and DDT combination therapeutic bispecific approach could be that leap forward that we need to make more progress within this field. So what we're asking for is basically funding to create this initial bispecific antibody. We have additional funding already in place to help bring it through preclinical studies. And to make this more realistic, we already have in vivo high affinity CDRs in place against MIF and DDT. Thank you.
Questions? Please.
Can you speak a little bit more about patient selection and what tumor types and what percent of those tumor types would have expression of both of these?
So there are multiple tumor types, head and neck cancers, ovarian cancers, hepatocellular cancers. We've also demonstrated the same clinical benefit in mouse models of colorectal cancer as well. And so typically all the immune sensitive cancers we anticipate to respond. It's really a tumor agnostic approach. A lot of cancers have upregulated expression of MIF and DDT.
Great. Thank you very much.
Thank you. Good afternoon. My name is Xiaolei Su. I'm a faculty at the Yale School of Medicine. Today, I'd like to share with you about my idea in developing a new version of CAR-T with improved antigen sensitivity. I'd like to start by introducing my team, which includes myself and my postdoc, Xinyan Zhang, student Fawzan Hashmi, and my collaborator, Sidi Chen, who pioneers in genome editing technology, and Iris Isufi, who is a clinician and the director for CAR-T therapy at Yale New Haven Hospital. So the challenge we're trying to address here is the current CARs have low antigen sensitivity, which means they can only target high antigen expressing cancer, but not low antigen expressing cancer. So the low antigen expressing cancer will escape the surveillance from the CAR-T. And for the high antigen expressing cancer, even they have the patient has very high initial response, like over 90%. But after 6 months, half of the patient will show a relapse. So basically the tumor grows back. And this is because tumor downregulates their antigen expression level below a threshold that can be detected by the CAR-T. To address this problem, we developed a called IDR CARs, which show the enhanced antigen sensitivity. And so now these IDR CARs can target low antigen expressing cancers. I think these IDR CARs will make a positive impact on cancer immunotherapy by expanding the target of CAR-T to these low antigen expressing cancer and also reduce the relapse frequency for these high antigen expressing cancer. So also I think this project presents a few innovations. First of all, these IDRs can be combined with CARs targeting a variety of different antigens, including CD19, CD22, or HER2. And these IDRs, because they are a protein peptide that can be fused to any CAR of your interest. So it's a pretty universal element. And second, these IDR CARs can induce biomolecular condensation of the CARs on the T cell surface, which increase the avidity of these CARs binding to antigen. So that's how it enhances its antigen sensitivity. And the biomolecular condensation right now is a really hot topic in biology. So we can learn a lot from the basic research, which will fuel the improvement and the design of new IDRs to improve these CAR-Ts. And last but not least, the sequence of these IDRs has nothing to do with CAR-T or T-cell signaling. So it serves as an orthogonal approach to improve the CAR-T and can be combined with other current approach, including introducing the co-receptor signaling or like cytokines to achieve additive or synergistic effect. Actually, there are hundreds of IDRs in the human proteome. Through some computational approach and wet lab, we can narrow down to these 3 IDRs that can robustly promote the condensation of the CAR on the T cell surface. And here I'm showing you that 2 of them, the IDR 1 and 3, represent by the orange and the purple curve that can significantly enhance of the killing of low antigen expressing cancer cells by the CAR-T as shown on the left. And also it enhances the survival of mouse in a xenograft model as shown on the right. Okay. With the support of this fund, I'd like to test the safety of these IDR CAR-Ts because I think it's critical to make sure that these new IDR CAR-T does not induce substantial cytokine storm and tissue toxicity, which I think is a key issue to be addressed for all the CAR-T therapy. And the goal here is really to identify the IDRs that can achieve a balance between a good tumor eradication and the tissue toxicity. So with that, I'd like to thank all your attention. Happy to take on the questions.
That was perfect. Question on the dozen.
What mechanism does the IDR drive increase?
Yes, so what mechanism drives the increased antigen sensitivity by IDR? So IDR is, it stands for intrinsically disordered region. So it's a protein fragment that can promote self-assembly of CAR to form these condensates. So you can think about it's a clusters on the T cell surface. So that will increase the avidity of the CAR binding to the antigen. So basically, the traditional CAR only have like 1 interaction site with the antigen, but by form the condensation, you have multiple actually interaction sites. So that's how it can recognize the low antigen.
Awesome. Perfect. Thank you.
Thank you.
Yingqun, you're up next.
Thank you. Hi, good afternoon. And so I'm a faculty at Yale School of Medicine, and I'm going to talk about treatment for cancer-induced cachexia. Cachexia is characterized by anorexia and body wasting. It affects 1.3 million of cancer patients each year, and in the U.S. alone, 80% of cancer patients will suffer from cachexia, and 30% of them will die directly from cachexia. No FDA-approved drugs to treat cachexia, and there are 2 in phase 2 trial. One is a curcumin food supplement. The other one is a peptide inhibitor, which targets a brain area, but the target area is in the very deep in the brain, and then probably you have to increase a very, very high dose to get in there. But what I'm going to talk about is ours will be very easy to get into the brain. And one in phase 3 trial, which is using progesterone analog, and but so far has found to induce severe blood clotting. we have developed a method to treat cancer-induced cachexia using a completely novel mechanism. And our method activates a subset of neurons in the hypothalamus called AGAP neurons. And cachexia is known to be associated with inhibition of AGAP neurons. When the neurons are inhibited, the TET3, I'm going to talk about the TET3 protein in AGAP neuron will increase, and then it will reduce appetite, increase energy expenditure, and increase stress. However, when the neurons are activated, you see the TET3 expression is decreased, and then you have increased appetite, decreased energy expenditure, and decreased stress. We have a novel small molecule called Bobcat 339, which can activate AGAP neurons, and we have a patent application with this small molecule and also its derivatives. The mechanism of action is that Bobcat induces TET3 degradation both in vitro and also in vivo in mice in AGAP neurons, degrades the protein. Importantly, when TET3 is degraded in the AGAP neurons, it activates AGAP neurons, and you want activation to increase appetite. We have validated our method in 2 mouse models. One is activity-based anorexia mouse model, and we show that a bobcat can correct 3 key features of anorexia by increasing food intake, maintain body weight, and decrease obsessive-compulsive wheel running. And also, bobcat can decrease anorexia-associated anxiety and depression, and decreases stress hormone like cortisol. Importantly, our molecule does not have liver toxicity and kidney toxicity. And we also have a ovarian tumor mouse model. We show that cancer in ovarian cancer increase TET3 expression and inhibit inactivate AGAP neurons, and our molecule can restore TET3 protein level and also reactivate AGAP neurons. We propose to use a mouse model, Lewis lung cancer carcinoma, LLC model, to further validate our small molecule. Why lung cancer? Because it accounts for almost 1/5 of all cancer deaths worldwide, and the LLC model is very well established, widely used animal model for cancer-induced cachexia. And we would like to have the 30K accelerator fund to perform efficacy study in LLC, and then to set up a stage for later PK and PD studies, and also toxicology studies of our method. The ultimate goal is to develop our method into a clinical, into a drug to treat cancer-induced cachexia. Thank you for your attention.
Questions from the judges? Anyone?
Have other people tried to drug this target in the past?
No, we discovered this just for the past 2 years. We published a paper in Journal Clinical Institute of Investigation and PNAS. So we were the first to discover the molecular target and cellular target, and also first to use this in vivo in mouse model.
What measures of potency do you have for the bobcat molecule?
Potency? Okay, so we have done this in drinking water and in intravenous injection and also peritoneal injection, 2.5 mg per kilo of mouse body weight, injection once a week, and then it has effect. And also because AGAP neurons are in the hypothalamus, it's very permeable to the blood things. So you can have a low dose and get in and then soak it up, and not like in the deep in the brain.
Perfect.
Thank you.
So that concludes the pitches. I'm not sure what the plan is for the judges' deliberation. Are we giving them time now because the award ceremony starts in 10 minutes?
You can follow me and we'll go to a breakout room.
Great. Thanks everyone for coming.
I think I told you. Yeah, I know, that's fine. See you enough. See me enough. Guys. And how, like, rational drugs to them, what they my latest. By the time I came back, he was like, yeah, I was just like, if I'm going to find a job, it's just going to be on me. I should be involved in your installation. That's big. That's all your innovation. Yeah, I think we've heard of it. I mean, the only downside of it, I guess that's why I mean, they have a Boston station now.
Somebody moved out.
So it's exciting. I'll get to meet a lot of people. Again, it's like, we're not anyways, my goal today was learn about what the new one I discovered is Yale Ventures. We've been trying to get it's actually pretty simple. That's really good. Wasn't there
tolerance, that may be very biologically active, even without the
he is an indication, he's a mutant, which is I'm still trying to like balance lab and yeah, so I'm gonna come back to the fall. I have my outline. I feel pretty good about it. I like every avenue I can. I just have unfortunately right? But it's very close to they are still suffering. Fine. Yeah, so have it right. This presentation you talk about I see. Wow. You have to make a direct right.
Sorry, I missed your presentation.
Do I have
co-founders Cardiobio. Did she go to Yale?
Induced inflammation, but cancer is cell and tissue specific. It's not like you get into the cell with the brain and pop. Not necessarily. It's somehow side effect. You know, like liver. And this is Cachexia and the
I had no figure in this lab. I have nothing to give besides wrapping up some labs. I'm so sorry.
We have all the undergrads.
based on racial design,
It's the tiniest like I just the past couple times I've seen him, but for a while, every time I signed these things, I thought they would shit the lab. Apparently.
You know, and the CRISPR chip sense tiny biosignal. And how much revenue did this
A couple of tens of thousands, maybe $200,000 for you this week. AstraZeneca, Bristol Myers Squibb, Being Therapeutics, everyone's kicking tires. The business model is not determined, but it's going to be a sense because we're going to buy boatloads of chips and then it's all about that analytics. So the most recent event, besides the prior company getting acquired, is huge.
And that prior company,